In this video, we will discuss about this CAR T cell therapy can cause oligodendroglial and myelin defects. In the previous video, we have discussed that CAR T cells has some negative impact in terms of this white matter formations. That we have seen that post CAR T cell therapies can cause white matter microglial formations in different CAR T cells models that this paper has used. Now we want to find out, or the researchers want to find out the reason behind this white matter microglial formations. So they hypothesize that it might affect this oligodendroglial formations or oligodendroglial precursor cells formations. At the same time, it will also affect these oligodendroglials. So they checked it in these different kind of CAR T cell models that they are testing. First, they checked it in diffuse intrinsic pontine glioma, and here they check the white matter oligodendroglial precursor cell formations, and they can see that in the GD2 CAR T on target therapeutic approach, they see reductions in this oligodendroglial precursor formations. At the same time, they also see reductions in the white matter oligodendroglials in this on target CAR T cell, GD2 CAR T cells models or this therapeutic approach. And similarly, we can see the difference in this immunofluorescence image in the mock T cells and the GD2 CAR T cell therapeutics for white matter oligodendroglial precursors cells, and also for this white matter oligodendroglials, the immunofluorescence image also. Then they checked it for the acute lymphoblastic leukemia CAR T cells. Here also they see the similar results that on target CD9 CAR T cell therapies cause reductions in these oligodendroglial precursor cells, and also it will show reductions in these oligodendroglials, white matter oligodendroglials in the CD19 on target CD19 CAR T on target therapeutic approach compared to the controls, that is mock T cells or GD2 CAR T off target. Next, they also check it for the third CAR T cells models, where they see the similar results that on target CAR T cell therapies cause reductions in this oligodendroglial precursor cells, but these white matter oligodendroglials and also these white matter oligodendroglials goes down in the on target CAR T CD19 CAR T cell therapeutic approach. But when they check it for immunocompetent murine melanoma models, they do not see a decrease in the white matter oligodendroglial precursor cells, but they see a decrease in this white matter oligodendroglials. And the aggressive CAR T cell model lines, they do not see a changes neither for these precursor oligodendroglial precursor cells and white matter oligodendroglials. And this is also in line with their results for the aggressive this is also line in the results for this rapid clearing osteocarcinoma. So this rapid clearing osteocarcinoma does not show any change in the white matter microglia. So, and this is also in line with their no changes shown for oligodendroglials precursor cells and white matter oligodendroglials. Those results are in the supplemental figures. When they check it for murine melanoma, they see changes when they use this chemotherapy, but they do not see any further changes when they use the on-target CD19 CAR T cell therapeutic approach. And similar results they have seen for white matter oligodendroglials, that they see changes when they use first chemotherapies, but they do not see any significant changes afterwards when they use CD19 CAR T cell therapies. Now they hypothesize that the decrease in this oligodendroglial precursor cells might be because of this myelin defect. So they took this transmission electron microscope images of these myelinated axons in the mock T cells and the GD2 CAR T cell therapeutic approach in this diffusive intrinsic pontine glioma, and they can see that a decrease in the myelinated axons in the MOCT cells it is high, but for the on GT2 CAR-Ts on target therapeutic approach it is low. This TEM images also shows this shown here, which has been quantified on the left hand side of the panel J, that for the MOCT cells we can see the more myelinated axons per micrometer squares, but for the GD2 CAR-T on target therapeutic approach we see a decrease in the myelin axons per micrometer squares. So all these results finally pointed out that why there is a decrease in the why there is white matter oligogia formations that we have seen in the previous video, that it is because of the decrease in this oligo precursor oligodendroglial precursor cells, and also a decrease in the white matter oligoglia, and this is because of disruptions in these myelinated axons. So if you need to understand more about their experimental results in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.